pure culture is crucial for accurate identification and further study of microorganisms, and streaking method is essential for obtaining a pure culture from a mixed population of microorganisms. There are several types of streaking methods used to isolate pure bacterial colonies on solid media. The main streaking types are quadrant streak method, T-streak method, radiant streak method, zigzag streak method, and continuous streak method. Today, we will learn how to perform T-streak method or technique, which is one of the most effective techniques to prepare a pure culture of bacteria. Welcome to this video. Let's get started. For streaking techniques, an inoculating loop is essential. It typically consists of a nichrome or platinum wire attached to a handle. In some cases, pre-sterilized disposable plastic loops are used. A Bunsen burner is also required for sterilizing the loop before and after use. To cultivate microbes, we need appropriate culture media plates, such as nutrient agar or selective media. Additionally, a mixed microbial culture plate is needed as the source from which we aim to isolate a pure culture. On the mixed culture plate, you can observe colonies of various microorganisms, differing in shape, size, and color, indicating the presence of multiple species. This is another type of plate that contains a single type of colony, which can also serve as a microbial source. T-streaking is one of the most effective techniques to prepare a pure culture of bacteria. First of all, flame and burn the loop in the Bunsen burner until red hot. Let the loop cool for a few seconds. On the bottom of the plate, draw a T-shape to divide it into 3 sections. Gently touch and pick a single, well-isolated colony from the mixed microbial culture plate using a sterile loop. Prepare a smear on section 1 using the bacterial cells collected on the loop. Flame the loop in the Bunsen burner until red hot and let it cool for a few seconds. Streak the section 1 horizontally using short, parallel strokes. Repeat sterilization by flaming the loop. Allow to cool the loop without touching anything. Drag the sterile loop from section 1 into section 2, streaking in a zigzag pattern. This spreads and dilutes the bacteria. Now, it's time to streak the final quadrant. This time we don't need to burn the loop. Without burning the loop, drag the loop from section 2 into section 3, continuing the zigzag streak. Expected result is dense growth in section 1, less in section 2, and well-isolated colonies in section 3. Close the lid of the Petri dish, invert the plate, and incubate it at 35 to 37 degrees Celsius for 24 to 48 hours to allow optimal bacterial growth. After incubation, carefully remove the plate from the incubator and prepare to observe the results. On the tear streaking plate, we can see numerous isolated bacterial colonies. There is dense bacterial growth near the initial smear. The first section shows a very few isolated colonies mixed with heavy growth, while the second, and especially the third section typically displays well-isolated colonies as a result of progressive dilution.